Now we're going to talk about the essentials of cellular metabolism because although it seems like torture when we're teaching you the stages of cellular metabolism, it's actually a pretty darn important topic when it comes to how our bodies work. Right? So, what I'm going to say is today we're going to focus on sugar as opposed to focusing on the other metabolites such as lipids and proteins. Those are also essential, but when it comes to when you're first starting anatomy, it's important to really make sure you know sugar metabolism because it's going to come up over and over and over again, right? So I'm going to take in sugar, otherwise known as glucose, into my body. And when I bring that into my body, I'm going to need to break that down. I need to break down all those bonds to be able to turn that into energy. What I'll do first is I'm going to go through glycolysis, which the term glycolysis is glyco for the sugar and lysis for break. So I'm breaking my sugar down. And ultimately, that's going to lead to having a molecule called pyruvate. Pyruvate it can be used for a lot of things, and it actually is a metabolite for many other parts of the cell. But when we're thinking about energy, right, this pyruvate will eventually be brought into the mitochondria. So we'll draw a little mitochondria membrane. And when it gets brought in there, we make acetyl-CoA. Acetyl-CoA, I want to point out, is not just used in cellular metabolism. We're actually going to be using acetyl-CoA for creating other signaling molecules. Used for some signaling molecules. It's going to be used for some other key chemical reactions. So in order to get the acetyl-CoA that you need for some of the most essential reactions in our body, you need sugar, right? So everyone needs to eat carbs. They're not the enemy, they are essential. Then acetyl-CoA can be used, and it's going to be used in this cyclic fashion called the citric acid cycle. Or the Krebs cycle, named after Dr. Krebs. In the Krebs cycle, we're not going to go through this today, but there are many, many stages, and each of those stages allow for us to use those metabolites for other things, right? So those metabolites that we make at each stage of the Krebs cycle don't always go straight into the next reaction. So as you go through anatomy and you go through physiology, there are going to be times where we're going to mention a specific enzyme or a specific molecule that actually uses one of these metabolites. But for what our purposes are, right, this Krebs cycle is going to allow for us to produce a lot of NADH and FADH2. These molecules, right, are essentially just electron carriers that allow us to go through the electron transport chain. Okay, we've got our little complexes. Okay, we've got 4 complexes, 1, 2, 3, and 4. And then we've got the ATP synthase. These hydrogens that are attached to NADH or FADH2 can easily pop off of them and move through these, so in NADH through 1, FADH to 2, and the electrons are going to eventually become stabilized through these complexes. They're going to be brought through. And then we're going to have this chemical gradient on one side of my mitochondrial membrane. One side of my mitochondrial membrane. And we want that gradient to be more in equilibrium, so those hydrogens can move down. And when it moves down, it spins my ATP synthase, and I create ATP. This is not the only place where we make ATP, but it's the main place where we make ATP. As you go through anatomy, make sure you remember where we are in each stage. And honestly, it would be good to look over what are all of the chemicals that are found in the Krebs cycle. We just don't have time for that today.